We're at the top of the time slot, so we don't have time for any questions, but there is one from the audience, so I'll ask you to address that after the fact.
That's fine.
Thank you once again. So we'll move on now to the penultimate speaker of this webinar, being Stacy Dee. Stacy is a research and development scientist at Labcorp with over 18 years of laboratory experience, including 12 dedicated to R&D. Her current work focuses on the development and validation of high throughput LCMS-based assays for small molecules. Today, she will present a summary into her latest for developing and validating a clinical assay to measure catecholamines through the aid of stable isotope labeled standards. Stacy, the floor is yours.
Let me go ahead and share. All right, can you see my screen?
Not yet.
How about now?
Yes,
Is that
perfect.
good? All right. So today I'll be talking to you about overcoming some of the challenges of plasma catecholamines assay development and validation. So the first thing you may want to know is what are catecholamines and why should we measure those? So this test is often used in conjunction with plasma metanephrines to determine the presence of different tumors and autonomic dysfunctions. When tumors are present, catecholamines are secreted in higher levels. When there are autonomic dysfunctions, they're actually reduced. They don't excrete as much. So structurally, catecholamines are, and let me see if I can get the pointer. So catecholamines are this group with a vicinal diol at the end. And then all the catecholamines also have an amino ethyl group right around here. And it's actually this vicinal diol that kind of separates the catecholamines from the metanephrines and makes it possible for us to use that mechanism to separate in the extraction phase. And you'll see some of that later. So we've had several technical challenges. Since we have to measure both low levels and high levels, that's challenging in itself, but I want to focus on how we used extraction, separation, and carbon-labeled internal standards, carbon or nitrogen-labeled internal standards to address matrix effects and drug interferences. And we used temperature to address stability issues both during collection and post-collection, and how we use metrology to show accuracy to a reference method so that we could verify an established reference interval. So the first thing we did was we looked at separation. Here you see a helix separation. On the bottom, you'll see a chromatogram showing that all three analytes co-elute. They also don't have very much response. So that was one thing that concerned us. We did a post-column infusion. The setup is here on the right. And we just teed in a constant flow of analyte into the stream that comes out of the column. And then we monitored that analyte concentration or that analyte response over time as we injected both an unextracted blank and a plasma sample. And what we were looking for was matrix effects and ionization differences. And what we see here is just that we're in a suboptimal ionization space with this particular separation. So we chose to go to reverse phase where, whoops, sorry about that. Reverse phase where we got much better separation. But if you look down here, I don't have the K prime on here, but this is almost at the solvent front where norepinephrine is eluting. So we needed more control because, as you can also see, in this trace that there's an area of suppression in plasma samples that you don't see in neat samples. So we wanted to avoid that, but no matter what we did with this separation, we couldn't move it out of the way. So we added 1% HSA into our reconstitution solvent, not into the mobile phase, but we added it to the recon solvent as an ion pairing agent. So that actually gave us a little bit more retention time, and it also gave us a little bit more control over the separation. So the next thing we did was we looked at the extraction. And at this point, we really needed sensitivity for dopamine because that one has the lowest cutoff. So here you'll see a slightly better recovery and slightly less matrix effects from the alumina plate. The weak cation exchange was comparable, but there was another piece. The hydrogen bonding that occurs with the alumina gave us a way to separate catecholamines, which are right here, from their metanephrines, one of the metabolites. So we chose to continue on with alumina. And then we wanted to keep optimizing to try to get more and more sensitivity for dopamine. We optimized the percent formic acid in the elute solvent, and we got to 5%. And then we optimized the elute volume using that new percent formic acid, and we got a 500 microliter volume. Now, this really was a balance of matrix effects, recovery, and evaporation time. So it wasn't just one consideration. Just because the 1000 gave better recovery, it also gave a little more matrix effects. So we had to take that into consideration. Another thing that we did was we had two choices of internal standards. On the top, you see the deuterated internal standards, anywhere from D4 to D6. And then on the bottom, you'll see the carbon and nitrogen labeled internal standards. These were all sourced from Cambridge Isotopes, and they kindly synthesized the norepinephrine internal standard. So thank you for that. That really helped us a lot. And on the next page, you'll see why we ended up going with those carbon labeled internal standards. All right, so on the left, the chromatograms are analyte and then internal standards. So here you'll see norepinephrine and then the carbon-13 labeled version, which almost perfectly co-elutes with the analyte. However, this D6 norepi does not perfectly co-elute. It's actually a little earlier in the window. So what does that mean? Well, we did another infusion trace and found that there is an area of suppression still with our new separation. And let's say we use the deuterated internal standard. What would happen there? So if you can imagine taking this piece over here and this being your analyte, you're probably going to be right around here, right? So it would experience less matrix effects than your analyte. And this is just one infusion of a small plasma pool. There are potentially many, many, many different samples that we would come across that could suppress the signal even more than that one. So that was the main consideration there. So we went with the carbon labeled. And on the next page, you'll see we have the same effect for epinephrine and dopamine, where the deuterated internal standard elutes before the carbon labeled, and the carbon labeled pretty much co-elutes with the analyte. All right, so this is our procedure that we validated. We have sample plus internal standard plus a stabilizer solution. So we're only using 200 microliters of sample. We send that through an alumina B SPE extraction. The LU8 comes off, it's evaporated, reconstituted in a 1% HSA solution or heptane sulfonic acid, and then it is injected onto the LC-MS system. And here's some key validation metrics. Now, it wasn't just development that had its challenges. We also had challenges in validation. So I wanted to talk about the drug interference, accuracy to a reference laboratory, and stability of patient samples, and the verification of a reference interval. So the first thing you'll see here are drug interferences. For the most part, we didn't have any drug interferences. These were the only ones that we saw. On the left, you see that L-DOPA interferes with norepinephrine at levels higher than 80. What it does is it raises the baseline, so you can't integrate the full norepi peak. In the middle, you see that M-DOPA contributes to epinephrine or interferes with epinephrine, and it does something similar. So anything above 200 nanogram per mil, you'll start to see an integration issue, and it's because of this baseline. It's a little more subtle with epinephrine than it was for norepinephrine, but it's still greater than 22% off of what it should be. And then dopamine, this one's a little harder to see. So if you follow the arrow down, the arrow marks where the internal standard is and where the analyte should be. And then this arrow is off on the side of the peak. And that's because you have pretty much a co-eluting interferent, an isobaric interferent, and it's massive at this point. So it looks like it shifts your peak, but you're just not seeing it because the analyte peak is so small in comparison. So what this meant was that patients could not take L-DOPA or M-DOPA. That's actually a rejection criteria, and it's consistent with all the other labs that are running this test. The next thing we looked at was stability, or instability rather. And what you see here is this blue line shows that in room temperature tubes, dopamine is not stable at all. So to stabilize our analytes long enough for us to draw the sample and then get it into the freezer, we had to use chilled tubes. These tubes were chilled on ice for at least 15 minutes prior to the draw, and then they were placed back on ice. And this is kind of a big thing to do if you're drawing a lot of patients. So let's move on to the next part. So we also looked at instability post-collection. Here you see that at room temperature, you have no more than 2 hours stability. Refrigerated, same thing, 2 hours. And then frozen, you have 24 hours. And then if you're frozen at -70 or less, you have 28 days. So what we took away from this is that samples can be stored only in a -70. So when they're poured off at the draw site and frozen on dry ice, they need to come to our lab and be placed immediately into the -70. Then after that -70, they can come out for 2 hours or less. And that's based off of this room temperature stability. So samples can only be thawed for 2 hours, and then they have to go right back into the -70 so that they don't degrade. So all the stability talk and the difficulty of collecting under those kinds of conditions, we were looking at how we wanted to do a reference interval validation. So CLSI EP28 says that we can do it 2 ways. The first way is de novo reference interval generation, in which you have to have 120 healthy donors. And then the other way is that you can verify an already established reference interval. In that one, you only need 20 healthy donors. 18 of the 20 must fall within that established range. But to do that, you have to show that your method agrees with the reference method, the one that generated that reference interval. So in deciding which one we wanted to do, we also considered the donor prep. So any donor that gave to our reference interval study could have no caffeine for 12 hours prior to the draw. And that was the biggest deal breaker of the whole draw. I had so many people say, I can't do without my coffee. So something as small as no caffeine for 12 hours really made this a little more difficult. So patients also can't be taking certain medications, high blood pressure medications, amphetamines for things like ADHD. And during the collection, donors must be seated for at least 15 minutes prior to draw. And this is actually a compromise. Supine is preferred. Like if you're in a hospital setting or something like that, your plasma catecholamine test should be done. Well, your plasma catecholamine draw should be done in a supine position. And so we settled with the seated because the established reference interval listed seated and supine. So we were going to try to establish verify their reference interval and then apply the supine if that worked. The other complication was drawing into chilled tubes and keeping on ice until the plasma was separated and frozen. So all these complications made it kind of a no-brainer as far as what you would choose. We obviously chose to verify and establish reference interval because we don't have the setup that's required to do a study this large with the ideal conditions. So the agreement with the reference method is shown here. You can see that we have good agreement with the reference method, good correlation. We had passing Bablock slopes that were between 0.9 and 1.1, except for the case of epinephrine. It had a passing Bablock of 1.124, so it's about 12% biased. So what we had to do is transform those ranges or those reference intervals. So the original reference intervals for seated and supine are here. So 60.4 was transformed into 68.8, and the 48.5 was transformed to 55.4. So after that transformation, we were able to go ahead and test our 20 samples, and no less than 18 of 20 were within those reference intervals. So we were able to successfully verify the established reference interval. We also adopted the supine reference interval. So some conclusions. In spite of the interferences, the instability of samples, and the patient prep, we were able to thoroughly validate an assay that was accurate and precise with low matrix effects and a successful reference interval and conditions that we could use to move forward with extraction and collection. So with that, I wanted to thank my R&D team members. The ones that are highlighted are the ones that gave a bit of their life to this project, and it's lasted for a very long time. So thank you very much to them. And thank you to Cambridge Isotopes, because without it, we would not have had our norepinephrine internal standard. So thank you very much.
Thank you, Stacy, for that nice talk. It's insightful always to see the work undergoing at Labcorp, both at the prep and the validation side for clinically driven applications. We have time for one or two, possibly three questions. There are three questions from the audience, so I'll go right down the list, starting with the first on the analytical column, geometry.
Mhm.
What reverse phase column did you use for the PCAT assay?
So that is actually back a few slides. I think I put it on the, so it's a kinetics, and I think I might be blocking it there. So you have a 100 by 2.15 micron kinetics biphenyl.
Yep, good. Thanks. The next question from the audience is what are your considerations on using catecholamine over using metnephrine in terms of underdiagnosis?
So typically, plasmametonephrin would be the go-to for tumors and that kind of thing, like the pheochromocytomas and paragangliomas. But when those are inconclusive, they come to the plasma catecholamines. That's when it's primarily used.
Okay. Thanks. Then the final question from the audience is for the PCAT stability, did you consider adding any preservatives to the collection tube?
So we couldn't add preservatives to the patient collection tube. However, we did add preservatives to the calibrators and controls to give them extra stability.
Makes sense. Well, thanks again for your nice talk. We'll move on now to the final speaker.